Okay, so welcome everyone to our TEO talk today. I'm Annemarie Wurz and I will be moderating today this talk series. And first, I say a few words about our Tropical Society for the ones who don't know it. So we are founded in 1987 and we are Europe's largest scientific association in the field of tropical ecology. And our society basically offers a platform to network, discuss recent findings, challenges, or developments in tropical ecology. And here we aim to foster applied research and to promote conservation of tropical ecosystems and also increase awareness of the importance of tropical ecosystems. So we have, besides this TEO talk series, we have also annual conferences. The next one will be in Czech Republic. You're all welcome to join. And we have also a journal called Ecotropica. It's open access and peer-reviewed. We have also grants and awards for researchers and also a tropical student group, which meets also monthly. And the society is open to everyone. So if you want to become a member, please visit our website. You can find the address on the lower right, and you can enroll here as a member. You can also join our student group. The first year is also free for non-members. Okay. So I welcome you to the TEO Talk series, and for the ones who haven't joined before, so we meet every second Monday at 3 p.m. And this TEO Talk series basically targets in particularly early career researchers and offers them a platform to present their research and to network. So therefore we combine a talk by an established researcher, today Pierre-Michel, for 30 minutes approximately, and then 1 or 2 presentations by early career researchers. At the end, we will have a discussion for a maximum 30 minutes to give everyone the opportunity to ask questions. And if you have questions, during the talk, keep them, and you can write question marks in the chat window, and Melanie and me will make sure that everyone gets to ask their question. Okay, so today we have Pierre-Michel Forget and Desamarie Fernandez as guest speakers, and both of them are from the Natural History Museum in Paris, and Pierre-Michel Forget is professor there and focuses on animal-plant interactions, especially seed dispersal and tree recruitment in tropical rainforest systems. And he focuses especially on rodents, but also frugivorous bats, primates, and birds. And here he combines taxonomy and ecology to better understand how trees are dispersed and radiated in Africa as well as America. So at the moment, he works mainly in French Guyana and Martinique and Southeast Asia. So, and now I give the word to Desamarie, a PhD student of Pierre-Michel Forget, to present herself.
Hi, hello everyone. You can call me Des. My name is very long. And apart from being a PhD student here at the Museum of Natural History in Paris, I'm also an assistant professor under the University of the Philippines, Los Baños, in Los Baños, Laguna, Philippines. So thank you very much for this invitation.
Yeah, fantastic. So I allow now Pierre-Michel to share his screen. And please, everyone, keep yourself muted throughout the talk, and then we collect questions until the end of the 2 presentations. So we have a shared discussion at the end.
Yep, thank you. Thank you, everyone, for being here with us. And thank you, Anne-Marie and Melanie, for organizing the TEO. I'm very glad to open it, although I was asked at the last minute to do it, but it's okay. We have some resources here. I don't know how many of you can see the title of my presentation here on this beautiful green background. It's written Nocturnal Frugivores and Carnivores of the Canopy Spotlight. Oh, it should be in the canopy. of the canopy in spotlight. So one main problem with nocturnal mammals, as you can imagine, and birds, is they are nocturnal. That means we are diurnal and we don't see them very well. We need a lot of equipment to observe them. And most of the time we do that from the ground. And on the ground, what we like to observe is what is very colorful, very visible, and easy to observe. Most of the time it is primates and birds, and also some ground-dwelling animals. To observe the nocturnal animals, then you have to change the way you're observing the forest, and then you have to move to a black and white world. And then obviously it's more obvious to observe these nocturnal animals once you have a night vision, for instance. Here we're going to talk today, it's about how we put some light on some obscure, less well-known carnivores that happen to be frugivores. And I'd like to co-offer this presentation with my former master students, Opal Coutan and Marie Seguin, as well as with Eric Guilbert, my colleague with whom we climbed a number of trees in French Guiana over the last years. So climbing the rainforest is quite very important, and especially to observe all those rainforest mammals that you don't see normally from the ground or barely see with a binocular. Even if it's in the understory, it's not that easy to see them very far away with a binocular, 10, 20 meters at least. And when they are in the canopy here in the orange area, it's even harder because sometimes it can be very high in the forest, very hidden behind the foliage. So now more and more frequently, people are using those camera traps here to photograph them and to have some evidence of their occurrence, of their presence. Of course, we don't see the same thing whether we use camera trap or binoculars. And I just wanted to mention here this beautiful graph from the community of Southeast Asia in Borneo, and I will come back in a minute about it. So here it's a big challenge for us who are diurnal primates. It is to observe nocturnal animals, whether they are mammals or birds, flying or not flying. So it's a main problem for us. So how do we do that? So now we have the chance that we have a number of technology, high tech, I would say, arboreal camera trapping. And it has been proven by my former colleague, Trevi Gregory. I say colleague because we've been together working sometime in Suriname. And she and a group at the Smithsonian, they developed the wonderful method and applied it to study the primate community that pass over a gas pipeline in the Peruvian Amazon. So she is really, I would say, the pioneer doing that. And she was followed by others. And actually, there's one PhD student, I think she finished, I'm not certain about that. She has been working hardly and it was difficult in Borneo to study those animals by day and by night, and doing the inventory of the diversity of this wildlife in Borneo, in log and unlogged forest. So it's a really hard work. It's a hard work. And recently we published with under the direction of Jennifer Moore, a paper showing how we should practice and use those arboreal camera in the trees, and just to remind people that it's not that easy. It's easy to go to the shop and to buy a camera, but it's not that easy to install them, especially whether you have to climb trees. So really, I encourage everyone to come back to these papers, to go to those links, and to read and read and read, and to train. And to train, why it's important to train? Because we are not climbers. I mean, obviously we are primates, but we lost some aptitude to climb into the trees. So most of our work in the tropical rainforest is from the ground. We observe the canopy, but we observe it most of the time from the ground. So we missed a lot. And to balance that, we need to climb and we need to go to the top, to the canopy. Many people have used many protocols to go there, but they're all very fancy, difficult, expensive, etc. And it's not always easy to go and not to disturb the forest whether you access the canopy. So here you have a beautiful forest of Nurac, French Guiana, where we have done a lot of studies but most of the time from the ground. And it's only recently, I would say, within the last 20, 30 years, maybe I started climbing at in 85 exactly, but it's really during the last decades that we developed the method, and using the method that are employed by arborists, as this fantastic person who climb here a tree, which is about 50 meters high. I'm taking the picture for the story. So what is climbing? Climbing is really going high in a canopy, and you can see here on the right a profile of the French Guiana forest at the river Desamarie, and you can see that a forest is. Our forest in the neotropics here is about 20 to 30 meters high, the canopy is, with some emergent up to 40 meters. So here we are exactly around here. And what is that? It is about 13th, 15th floor building. Here it's a 10th floor building, just to give you an idea. So you have to climb there to install camera where most of the animals are foraging. And that is not easy. So installing camera, you can also go and read that paper that was published some years ago in the review in the journal Ecotropica. Climbing trees require a lot of expertise and a lot of training. It is really crucial that you take a lesson, and we have been developing method with the arborists who work with us and develop method to better climb and also to climb safely. Because what is really most important is not only to climb, so to go there, observe, and go back and do that quickly, I would say, and safely. So we can do it now. It's safe, and believe me, it is really safe. I'm here to talk about that, and you can see me here on the emergent. We are about 40 meters above the ground, installing camera trap, and it works very well. It just requires some training. It's also required to know more about your camera, the camera traps you're using. I'm not going into more detail here, but you can go into the literature. There's links here. You can follow them. And all these will give you a lot of advice on how to use them, and also which equipment to use and what for. And definitely, the most important for everyone is to have a lot of camera, and to follow them, and to record, and to save the data, etc., etc. So whether it's easy to do that on the ground, I would say easy, somehow easy. That's not very easy when you decide to do that in the canopy. And also it's even worse when you are doing that in tropical rainforest, where unfortunately we don't have all these wonderful high-tech to access our camera and to record all the data that's been taken on camera using satellite or all these radar, etc. So we have to do it by ourselves. That means once you install a camera trap, you have to go there back and return them every month or every 2 months, depending on the kind of camera and battery you have there. So, but setting camera is not only the most important. What you must know, I will say first, is you must know better of your forest before setting your camera. And this is what we have done about 10 years more, 20 years ago. We started to know more. We started to know more about the forest, and especially about the fruits and flowers that were consumed by frugivores and folivores, etc. And we're going to focus here on this species, Pierre-Michel, during the rest of this talk. And just to mention some beautiful flowers I can see from the ground when it's open, but when it's closed forest, what you have in hand is only some flowers on the ground, some fruit on the ground, or some fruit you may collect here with strap, fruit that you will collect, dry, and then use and analyze in a lab. So from this, the 10-year, this decade of data we collected, we published several papers about the phenology, papers that were led by Natalia Norden here and by Hiren Mendoza. And what we've learned we learned a lot about the phenology. And what most important, we learned about the irregularity of flowering, of fruiting. And that is the most important, I will say, when you want to study animals in a canopy, because Whether you're going to set your camera, you also want to know how much food is available for them. And here you can see on the left that fruiting is very irregular. Even some years are totally lacking fruiting. Licania, Manicera, Sapotacea, that is a mast fruiting species. So when we as ecologists studying the interaction between plants and animals, and especially the fruit, the interaction between frugivores and fruit, what we want to do is also to study and to invest in trees that are the most, I would say, efficient in terms of data, in terms of analysis. And so we focus on this group of Virola micheli, the nutmeg family, because it is very regular, as we observe after 10 years of study that were summarized by Irene Mendoza here. And this three species is quite a good candidate to study interaction between plants and animals. Another one would be the Manicera here, Sapotacea, but because they are mass fruiting, then it's very difficult and very difficult to set cameras because you don't even know whether the trees are going to flower and fruit every year. I mean, they are not going to fruit every year, but you don't know exactly when they are going to fruit. So we focus on this group of Virola, not only because they are annual, but in the literature, there's a lot of information about Virola. We know from this pioneer paper by Ankao and Gay van Kerkhove that the trees are good fruits for primates and birds. And that is what we are interested in most of the time as a researcher. We know from that study that most of the fruits and seeds, especially, were swallowed and dispersed by birds, toucans, as well as by spider monkeys. We also know from that study that was carried out in Panama that most of the trees were barely visited by nocturnal animals. And I just here highlight in red the remark of the author that after 308 nights of observation, they barely see any nocturnal frugivores in a tree, except that once they observed one kinkajou that was sleeping on a branch of the tree. So from that, we decided that probably the kinkajou were not so important as frugivores and disperser. And when we studied the dispersal of seeds and seedling of Virola Coate in Nurag with Daniel Sabatier, and plotting the seedling around trees, between two trees here from A to B, we observed that seedling were dispersed, and this was very correlated with dispersal by spider monkeys. We still have a good shadow of seedling around the trees here. And from the data, from the observation by Daniel Sabatier, it was clear that most of the fruits were removed by toucans and spider monkey, and Potos flavus, carnivore frugivores, was only responsible for 3% of the dispersal. So in the end, when you invest your time in a canopy and you study the animals, you're not going to pay too much attention to nocturnal animals because clearly from the data we had at this time, they are not so frequent. So you invest most of the time to study them by day. And it is what we done later on. However, I would like to remind something that is quite very important. At Nurag in the early 90s and earlier in the 80s, Didier Julien Laferrière observed kinkajou at two forests, one at Piz de Saint-Elie and another at Nurag. And he did a very interesting study. Unfortunately, he did his study studying a signal. He studied only a signal. That means he followed the animals that were equipped with a radio transmitter here. And when you follow them, when you follow a signal, you don't really see the animals. But however, his study showed that the animals were very active over his own ranch, mostly active during the morning and at the end of the day. And I square here the time frame during which William Glantz was arguing that most of the nocturnal mammals were active when most of the nocturnal mammals were active. That was during the early hours of the day. And that wasn't a reason why Ankao probably haven't seen them, because in fact, we will see later that during that period, the animals are just moving from their roost, moving to the fruiting tree. So when you're studying them early in the night, we are missing a lot of their activity. So the question is and remains, what do they do during that period? And it is what we're going to observe very soon. So the kinkajou, the nocturnal frugivores carnivores, has a home range about 10 to 40 hectares in French Guiana. It can be a little bit bigger elsewhere. And they visit the different trees during the day, moving from the roost to the fruiting tree, and male and female interact. And they forage in the forest most of the time above 20 meters in height. That is really high in the forest. So even at night, it's very difficult to observe them. And Didier Julien-Ferrier followed a signal, in fact. What is most important from the work done by Didier is that the Virula micheli, a species that we are particularly interested in, was only barely visited by the kinkajou at night. Here, only one tree was visited. And for the little story here, he mentioned in his paper this little one with an A, that was seeds were collected in festus and the dermer sample. So there was one sample where he observed the animals that dispersed the seeds. And in fact, I was with Julien Laferrière when he did this observation in a field at Piste de Saint-Élie. So that's just for the little story, or the story is his story. Anyway, so apparently, definitely, kinkajou was not really a fan of the Virula micheli. And later on, he studied them at the station Nurag. And out of 41 species of fruit that were listed as consumed, 3 Virula were, however, consumed. Still, during the short period he did his work, the animals were not so frequently visiting the trees. So I just want to mention, however, that another study has been done in Panama, that also showed that kinkajou were not frequently visiting the Virula micheli in this forest. Just to mention that for people who are more interested in frugivores, carnivores, and I really like that picture of Roland with a kinkajou, and here's a picture of the kinkajou eating the flowers, picture by Christian Ziegler. So with that in mind, we did a lot of study. I say we, student Sandra Radiarizon analyzed interaction between arboreal frugivores and birds, and most of the time she worked by day and observed how much of the fruits and seeds were removed, how much of trees, how many trees were visited by different animals, etc., etc. She did a very great job in the early 2000s. And from that thesis, I'm going to focus on this paper that we published in Tropical Conservation Science, comparing the proportion of primates and birds that were visiting the two different Virula aquatae and Virula micheli. The Virula aquatae has a larger fruit, the Virula micheli has a smaller fruit and the smaller seeds. And she observed that there was a difference in the percentage of larger and smaller toucans. And she observed that ateles were equally abundant in both species. But something should be striking here to you. It's what about the nocturnal animals? What about kinkajou? And in fact, indeed, based on the literature, we have neglected the role of kinkajou in those trees, and not only the role of kinkajou, but the role or the impact of other vertebrates in the trees. So with that in mind, we are using this species as a model tree to evaluate how much of the forest has been disturbed by hunting, by logging, or by construction of road. And we use the fruits from the ground to evaluate how much of the fruits has been removed by arboreal and terrestrial animals. So here we base most of our analysis on the fact that normally the fruits are removed by frugivores, diurnal frugivores, mainly primates and birds. But there's I would say, something we totally don't know is how much of the fruits could have been removed by nocturnal animals. And so while doing those studies, former student Olivier Boisset and ourselves, we compared the diversity of animals and the species richness between two forests, one control forest, the Nurag, and another, the Montagne de Caux, that had been disturbed. And we observed not much difference, except that spider monkey was missing in the Caux forest. However, we also observed an increase of number of toucans in the disturbed forest compared to the control forest. So what are the consequences for removal? We might expect that licking the spider monkeys and howler monkeys, you would have a less dispersal. But in fact, we found the totally opposite. In the two forests, we found a very comparable level of seed removal, very high, above 80%, up to 95% here at the two forests, the control and the disturbed forest. This other forest, I won't detail here, it's a fragmented forest where most of the animals are gone anyway. But most important, we observed that the type of fruits on the ground was different. For instance, for within Mira Micheli, there was a greater proportion of fruits that were totally open and in a controlled forest, whereas they were still sealed at the base in the disturbed forest, where we expected less animals to remove the fruit. So with that in hand, with that knowledge, there was a problem, because despite the difference in the community of frugivores, the same population, not the same population, but despite the difference, we were still observing a lot of removal and a high percentage of removal. So there was two hypotheses. One is that there's possible compensation by toucans and the other primates that will remain in the forest, such as, for instance, capuchin, or maybe the Pothos flavus, kinkajou, will be more active in those trees when the other consumers are not present. So we set a number of experiment and observation in at the new field site at Nurag, at National Road 2, which is west of French Guiana. And there we install camera traps on the ground as well in the trees, close by the road, in order to observe the community of frugivores that were still remaining despite the road and despite the obvious effect of hunting along the road and into the forest on both sides of the road. Here, what we can see here, it's ecological corridors where most of our studies have been carried out. So there's about 13 ecological corridors, and we work at 7 of them. So here what we have done, with the help of Eric and other assistants and professional in tree climbing, arborists, we set camera very high in the canopy, up to 30, 40 meter high. And here we can see Opal, our former master student, who learned how to climb trees and to install cameras. And there we start to observe, well, the canopy. Of course, everyone told us, oh, it's a lot of work, you don't see anything, it's a lot of leaves are moving, etc., etc. It's true, it's true. It's obvious, as you could see on the video that I'm going to play again. Leaves are moving, there's wind, and most of the time there's no animals. So it is what you have to be aware of. But sometimes you might be very, very, very lucky. And I'm going to play it again. And this day, that was exactly the first week we set those camera. After setting the camera in a virula tree with fruit, as you can see here, still some fruit, within one day, we observed a kinkajou that was defecating seeds from the tree. So we were quite very excited by that observation. It was close to the road, and we thought, well, maybe we missed something. So the year after, with our crew, we started to install more cameras into more trees, and we start to observe a lot of kinkajou in those trees. And play it again, because I'm sure you like them very much, as we do like them, these little kinkajou, carnivores, frugivores, and here foraging into a fruiting Vera micheli tree. You can see the fruits here. So with that in hand, the two master students, Opal and Marie, we work together and we published these two papers recently this year in Frontiers and Scientific Reports, analyzing the result of this camera. So one of the main observations that in accordance with previous study, we observe halo monkeys, capuchin, toucans, Penelope marai, guans by day. But most important is that most of the time we also observe diurnal mammals that were foraging eating the fruits like porcupines, spiny rats, opossums, here filanders that has been also studied by Didier Julien Ferrier. But most important was that the most frequent animal observed in those trees was a kinkajou. And here I plot the red area that's been considered as being the most important for animals and for nocturnal animals foraging during the day. And you can see here that, in fact, whether they forage more in the beginning of the day, then they spend most of the time in the Vera in our study, and all Vera were very distant apart. So I play it again because I think it's so fun and so great. So kinkajou, in the end, appears to be one of the major consumer of Viola trees. So the next question now is whether they were more abundant because there was a lack of spider monkey, or whether it was just because there were because there's more resource available, so they are more frequently foraging into those trees when you are missing the other consumer, or maybe it's just a question of occurrence of kinkajou, and they do indeed are more frequent in Vera tree. It's just because we totally forgot to observe them during the fruiting season, because we were mostly focusing on diurnal species like capuchin. Here, spider monkey that appeared to be very late visiting the tree at 7:30. I think it was almost going to bed probably, and here toucan. So now with our team, we are continuing the story, and we are going to focus on the same trees, more climbers also, and more camera traps in order to compare the activity of carnivores and diurnal nocturnal carnivores, frugivores carnivore here, kinkajou, with the other normally diurnal frugivores such as spider monkey, capuchin, halo monkey, toucans, and others. So just to end, saying that maybe we don't see what's happening there, but definitely something is happening in the tops, and we should not forget about it. And thanks to those cameras, we can really see what is not observable from the ground with our lens, with our binoculars. So all these images, of course, they have to go somewhere, and we just loaded them at agouti.eu platform that is managed by Wageningen University, and so it's very useful. Here we have all our data that are saved somehow, and our sites are here listed. We have all the animals, number of animals identified and observed there. Just to show you how the platform is, we have here two capuchin in the tree that come to visit the Viola Quate, and one is here. Oh, I passed my 30 minutes. I'm almost done. And here, of course, another one with the two wonderful kinkajou. And you can see here, they are not really competing, except that they are competing for food. One being eating fruits during the day, the other during the night. And finally, I want to also mention that all the data we are gathering with those cameras can also be used by students. And actually, one Arthur Haas is using our data to study the evolution of the prehensile tail among Amazonian mammals. And another student in Estación Biológica de Doñana, Iago Ferreiro Arias, is studying the impact of deforestation, degradation on the community. So all those data can be used by many more persons. So really, thank you, everyone. Especially, I would like to thank my colleagues in my team because it was a lot of fun to do that work, and it's not finished. And we are going to continue this year, well, next year, in fact. And I would like to thank you all and for your attention. Thank you also all our sponsors, all our colleagues, our institution, all the donors who have contributed to make this study possible in the last 3 years since we started in 2018 with first test. And this, for instance, this last picture of the toucans was taken during that period of test that lasted for a week. So we decided it was very worth the time spent into the tree. And you can see here the fruits are still green, and the toucans is probably observing whether they are going to mature. So we have more data about birds, about nocturnal animals, and if you want to contribute to help, I encourage you to contact me either by Twitter or by email. And also encourage you to go to visit that YouTube where we have more information about the method and the protocol. So thank you very much. And I finished here. I'm ready to answer to your question. Thank you very much.
Thank you, Pierre-Michel. So we directly continue with this, and we will have a joint discussion after these two talks. Okay, so hold on with your questions, and I invite Des to share her screen.
Okay, is it okay?
Perfect.
Okay, thank you very much. So for my talk, it's going to be about my camera trap survey in Puerto Princesa Subterranean River National Park in Palawan Island, Philippines. So just to give a short introduction, so the Philippines is in the Oriental Zoogeographic region, right here in Southeast Asia. And specifically, Palawan is in like the crossroads of many different biogeographical realms. It's actually more closely related to the Sundaic zone than the oceanic Philippines. And in the Philippines, so this is an archipelago divided into different faunal regions. And these faunal regions are called Pleistocene aggregate island complexes, which were formed during the last Great Ice Age when the sea level was lower by 120 meters. And a part of these Pleistocene aggregated island complex is the Greater Palawan faunal region, which had shallow island connections, resulting in their similar faunal compositions. Right there. So that's the Greater Palawan faunal region. And carnivorans are from class Mammalia and order Carnivora. And there are 8 species in the Philippines, and 7 of them are found mostly in Palawan. In the rest of the Philippines, there are only 1, 2, or 3 species, and all 7 are in Palawan because, as I mentioned, they're more closely associated with the Sundaic faunal region area. And these carnivores, when you think about the word carnivore, you would think they are all carnivorous, but they actually have varied dietary preferences. For example, you can see here a picture of a common palm civet, which is omnivorous to highly frugivorous. And so they provide valuable ecosystem services such as seed dispersal. However, they are threatened with hunting and habitat destruction. So of the 7 species, you will find them here. So this is the common palm civet or Paradoxurus philippinensis, the Malay civet, which is least concern, the binturong, which is vulnerable under IUCN, the Sunda leopard cat, the Asian small-clawed otter, which is vulnerable, the Palawan stink badger, which is found only in Palawan, the Palawan faunal region, which is least concern, and the collared mongoose, or recently reevaluated as Urva semitorquata. So for the common palm civet, Sunda leopard cat, and collared mongoose, the reason why they don't have IUCN category yet is because they were recently either elevated to their own species or recategorized into a different species because of recent taxonomic studies. So this study will actually provide more information for the further evaluation of their conservation statuses. And the main topic of my thesis is ecological niche partitioning, where so competing species can use the environment differently. So the partitioning of niches help similar species avoid direct competition. And according to Chesson, the coexistence theory says that niche partitioning is a stabilizing mechanism with 4 axes. And these are resources such as food, time, space, and predators. But since carnivores are most likely like at the top of the food chain, we can take out predators. So the conceptual framework of my entire thesis is looking at the spatial, temporal, and dietary niche partitioning of carnivores in Palawan. But for today, I'm just going to focus on the camera trapping part, where we explore their abundance and other factors more in the spatial realm, and also a little bit in the temporal realm. So for the methods, the study site is Puerto Princesa Subterranean River National Park, which is 222 square kilometers and is a UNESCO World Heritage Site. So this national park contains many distinct forest types that are representative of other forest types in the entire island of Palawan. That's why I chose it as my study site. And it's famous for this underground river or subterranean river that is one of the most highly trafficked, the most highly, where the most tourists actually go to in Palawan Island. And Palawan Island is actually a big hotspot for tourism in the Philippines as well. So this is what the national park looks like. It's mostly forest land with some other land classifications, mostly residential, a little bit of farmland as well. And the forest here looks like this. So you can see there's a lot of exposed rock because there's a lot of forest over limestone. You can also see here some coconuts, bananas, and cashew trees planted in some parts of the national park along the edges. And also there are mangrove forests present. So for the camera trapping method, so I set cameras at ground level, 20 to 30 centimeters from the ground. And in my next field season, I will be setting them on the canopy 5 to 20 meters from the ground. And these will be set to record data for 2 seasons for 6 months each. And the locations of the camera traps are distributed on a 1 by 1 kilometer grid. So this is what my grid looks like. So you can see here, there are 19 camera trap stations and a big empty space here, which you might be curious about. Well, that's because the big empty space where I was not able to set up cameras is because that area was like completely inaccessible because of a recent typhoon that happened in the national park. So this happened in December of last year. And for the camera trapping, so I was there in March to April, and I set up 19 camera traps on the ground level, which was regularly checked by the National Park Rangers. And in July to September, my assistant was able to retrieve the data, and there were 11,949 photos with a trapping effort of 1,812 trap nights. So the average working duration for the cameras were 4 months with a minimum of 2.5 months and a maximum of 6 months. The image processing was done through agouti.eu, which was also already mentioned by Pierre-Michel. And this resulted in a total of 1,129 detection events, 184 of which are carnivores. And for the results, partial results, there were a total of 26 species that were detected, 19 of which were identified to the species level, and 7 are currently unidentified, mostly small birds and lizards. And of all the 19 identified species, the most abundant in terms of absolute frequency is the long-tailed macaque or crab-eating macaque. And next were rats of the genus Ratus and the squirrels and the Sciurus juvencus. And if we just take the absolute frequencies of the carnivores, the most abundant is the Palawan stink badger, followed by Paradoxurus hermaphrodistus, the common palm civet. And after that, we have the collared mongoose and the Sunda leopard cat, and 1 detection of the binturong. As for the relative abundance indices, this is the number of detections per trap effort times 100. The results are similar to the chart before, with the Palawan snake badger and common palm civet having the highest relative abundance index of all the carnivores. So just to show you a little bit of the photos that were taken. So here we have the binturong. You can see, so the binturong is primarily arboreal, so it's actually quite rare to see them on the ground, but we have 1 photo of an individual here. Another one is the collared mongoose. You can see him there behind the bushes. Another one is this lovely common palm civet, seemingly trying to catch an insect in this photo or something. And the Palawan snake badger, it's an endemic species that was the most frequently encountered in the camera traps. And then my favorite, the Sunda leopard cat. You can see him right here passing by the camera trap. Unfortunately, out of the 7 carnivore species, only 5 were recorded, so I wasn't able to detect the Malay civet or the Asian small-clawed otter during this season of fieldwork, but hopefully we'll be able to detect them next time. So for the other species recorded, so the most common one, of course, is the long-tailed macaque. You can see here a very handsome male looking at the camera. And also the Palawan bearded pig, which is an endemic species. Another endemic species is the Palawan peacock pheasant, which is actually the symbol of Puerto Princesa city, where the national park is located. So it's culturally very important to the area. And the Palawan porcupine. 2 more endemic species are the Palawan tree shrew and also the northern Palawan tree squirrel, which is the 3rd most frequently encountered species. So here are some preliminary insights from what I've done so far. So it seems that the most frequently detected was the stink badger and the palm civet. In another similar study in Cleopatra's Needle critical habitat, which is also in Puerto Princesa on the opposite coast as the national park. The common palm civet was their most frequently detected carnivoran. However, their design is a little more different and they're more focused on getting as many species as possible. So they actually baited their traps with pig's blood to increase detection, while my traps were unbaited. Also, for the temporal partitioning, it seems like the collared mongoose is more often detected in the daytime, while the other species were mostly detected during nighttime, of course, with the caveat that our sampling size currently is not as large to make like a conclusive statement on this. So further studies needed in more habitat types, elevations, and forest strata, such as the canopy, And for our next season of fieldwork, I'm going to deploy 30 more ground camera traps and 10 canopy camera traps. This will be in October, and it'll be in previously inaccessible areas. So now I'll be able to go to the areas that I wasn't previously able to go to. And the spatial niche analysis, I'm going to use occupancy modeling and species distribution modeling. For the temporal niche analysis, it will be generalized linear mixed models. And I will also be collecting scats or fecal samples and look at them through DNA barcoding and visual fecal content identification. So that's all for now. I'd like to thank the following institutions for funding and supporting my research and also my studies here in France, and especially to the National Park Protected Area Management Board. Okay, thank you very much.
Thank you, Des, for these wonderful photos and insights to your PhD. And now we can open our round of discussion, and everyone is welcome to ask questions. You can post them in the chat or raise your hand, and Melanie and me will make sure that everyone gets their chance.
It would be good also to see faces of people.
Yeah.
That would be nice.
Yeah, feel free to turn on your camera.
You're...
Okay, we have a first question by Yev.
Can you hear me?
Yes.
Yeah, thank you very much for presentations. Actually, I have a question to this. Namely, I couldn't understand it. It was a little bit too quick. You mean that your cameras were working or worked for different number of days at different sites, right? Or for different number of months? Or didn't understand it correctly?
Yes. So they were installed around the same 2-week period, but they stopped working at different times because of perhaps the battery or the failure of the camera traps. So the minimum was 2.5 and the longest one was able to run for 6 months. The 6-month one was because the area where I set it up became inaccessible for my assistant because there was another big storm. It's quite a big problem in the Philippines to have these typhoons all the time. So it took her a while to retrieve the data, and it was 6 months of data. Yeah.
Okay, I see. It means that if you would compare the data between sites, it would be difficult to do it, right? Or are there any techniques to somehow equalize this difference? I'm asking because I already had such a problem in my master thesis, and I'm going to collect some camera traps data in the quite near future, and so it's interesting how people deal with such problems.
Yeah, so there's a 2-month period where all of the camera traps are working at the same time. I'm still going to look into which is the most appropriate thing to do to take all of the data all at once, or just take the period of data where all camera traps were running at the same time. So yeah, I can't answer it right now, but I might be able to get back to you in a few months when I figure it out as well.
Thank you very much.
Thank you.
I have a follow-up question to this. Do you have already some guesses what kind of differences you expect between the canopy and the ground camera traps? And also the different like forest and the buffer zones? As you said, the vegetation is quite different.
Yeah, so there are some studies already in Borneo, as mentioned by Pierre-Michel, and also some studies in Palawan from other organizations. But for example, the Arctictis Benturum Foundation, they only did an analysis of arboreal camera trapping. So for their data, they focus more on the binturong or the bear cat. So they mostly got that species and also the common palm civet. But for the ones that are limited to the ground, I would expect the stink badger would be limited to the ground because they actually burrow dens, and they to get insects and other animals, they would dig into the ground. So they're more of a ground species. Perhaps also the leopard cat, but they can climb. So that's going to be like, we're going to see how that's going to go when we do the camera trapping. As for the buffer zone and core zones, we expect that from also a previous study that I did for radio tracking of leopard cats. Leopard cats are quite generalists in their spatial niche. So they're not super dependent on forests. They can go into fruit orchards, coconut plantations, and other areas like that. But the two animals that I wasn't able to see, so the Malay civet and the otter. So the otter would be limited mostly through waterways. So that could be their niche. And then the Malay civet is complete mystery to us right now, where they're going to be. So I'll keep you posted.
Fantastic. Okay, any further questions? You can also write them in the chat if you are unable to speak with your microphone.
Oh, there's some question here.
Yes, you will get notification on Twitter and our website or our email if you enroll to our email list about when the recording is ready. I have a more like general question or about the kinkajou in specific to Pierre-Michel, because it seems like it's quite an important seed disperser. And what does the literature, or what do you know about like how many tree species depend on the dispersal from the kinkajou, and how far can they actually disperse these seeds? I'm not an expert on kinkajous at all, but I'm wondering where they stand in this complex seed dispersal network.
Yeah, thank you, Anne-Marie. For the number of species, so far the list of the number of species of fruit species end up to 40 species. However, we have to remind that this study was based on only one family. It's a couple, a male and a female, although they don't live together, but they interact a lot. And it's only from one site or two sites in French Guiana. So it's very a small sample of information. We listed the literature, and we found some more information. And with Marie Seguin in the paper published in a scientific report, we compare the size of the seeds that were dispersed by Pothos flavus, by kinkajou, with those dispersed by other primates. And it appeared that the size of the seeds were very comparable to those dispersed by spider monkey. If you consider the home range of dispersal of kinkajou is about, I mentioned that here in the presentation, 15 to 30, 40 hectares. They don't, I would say, they don't visit that many trees, fruiting trees in such a small area, even because there's a high diversity and the number of trees in fruit are very spaced apart. So if you consider just the home range, indeed, they would not disperse a large number of seeds. But whether you consider the entire surface of the forest and the accumulation of home range of different groups and families of kinkajou, then you may end with a much larger number of species. So compared to spider monkey has a home range of 250, 300 hectares. So a group of spider monkey, which is about 10 animals, can visit far greater number of trees than a single family of kinkajou. But if we had several families, 10 families of kinkajou, probably they may be responsible for dispersing a much greater number of species. Regarding the distance of dispersal, compared to a spider monkey or even to a capuchin, indeed, spider monkey and capuchin have a large home range. So they travel long distance. So they can disperse seeds on, for instance, along 200, 400, 100, 1,000 meters, 1 kilometer, up to 4 kilometers for a spider monkey, for instance. Whereas kinkajou, small size, 3 kilos, and a home range of 10, 20, 30 hectares, they would disperse seeds over 300, 400, at least 100 meters. So they would not disperse it as far as spider monkey or Cebus as capuchin. However, if you accumulate all these little animals over the forest, then kinkajou might have a role that is comparable, I would say, to other animals like spider monkey or capuchin or even howler monkeys. However, we have to consider one thing I didn't mention in my presentation here, is that kinkajous are frugivores, but they are carnivores as civets. What does it mean exactly? When you are a primate, a frugivorous primate, you have this little thumb that is very useful to manipulate. So a primate can open a fruit before it is ripe, whereas the carnivores only have claws like this, and it cannot really open the fruit if they don't handle the fruit. They really grab the fruit. We observe that with the pictures. They grab the fruit, they grab the branches, and they swallow the fruit when they are open. We don't think they open them. We haven't seen that. So there's a big difference. There's a big difference, and the difference is that the spider monkey, capuchin, howler monkey, by day, when the fruits are opening with the sun, they may reach the fruit before the kinkajou. So that might explain why, in the end, there's less fruits available for kinkajou. So what we have to observe now is whether there's a difference in the frequency of kinkajou in forests with primates compared to a forest without primates. And maybe when you are losing the primates, kinkajou, as well as many other carnivores that are frugivores, would be an important animal at dispersing seeds in forest, and would then play a complementary role or a compensatory role as seed disperser, which has been largely neglected so far.
But they also compete with each other, because you mentioned that in some parts you observed there were no spider monkeys, but more kinkajous.
That's really new, in fact. I think the spider monkey was apparently very late, and it was, and the kinkajou was just starting its day. So they might not, I will say, they might not interact day and night, but they will compete, I would say, indirectly, spider being reaching the fruit before the kinkajou does.
Okay, yeah, thank you very much, Pierre-Michel. We have another question in the chat from Jeffrey, and he's asking if you can recommend affordable but high quality camera brands that people who would like to do camera trapping could use.
Well, that's a very interesting question, but there are several papers that are reviewing the cameras, the pro and the contras. Pro will say is the quality of camera and also what you want to do with your camera. If your camera is just to set in the understory in a dry area, well, then maybe you don't need a very expensive camera that will be more adapted to wetter conditions and to hotter conditions. Just as, for instance, behind me, when you are in the rainforest at 40-meter high, temperature can be really high there, and the rainfall can be really also deteriorating your equipment. So I'm not going to promote one brand or another. I just want to say that we are using only one brand, and you can see the brand label in our pictures. But there's some good cameras at a lower, I would say, a lower price. But you know, you have the camera. Someone said you pay for what you have.
Okay. Yeah, it's always complex with these choices and always depends on your objective.
And it depends on your money also, because it's
On
very
money.
expensive. It's very expensive. So just to give you an idea, the camera we're using, the one that Des is going to take to the Philippines, it's 5,000 euros for 12 cameras, including all the equipment necessary. That means memory cards and batteries. So that's a huge amount of money. But in the end, after 2 months, 3 months, your camera are still working, and you have good data. So it's a question also of investment.
Yeah, you really have to look at your sampling design and your budget, and then decide what kind of camera trap quality and price you need to target. Yeah, and unfortunately, if I assume if you're working in the Philippines, you also have to think about the shipping cost of importing camera traps, because they're not readily available in the Philippines and other countries.
Melanie has a question.
Yeah, thanks for these great talks and for sharing these wonderful pictures of elusive species and videos. My question is very much related to what Anne-Marie asked before about the role of kinkajous and C-dispersing primates. How are the kinkajous basically distributed in disturbed forests, or also at the edge of forests? So can they be very important as seed dispersers in kind of regenerating processes? What do you think?
Absolutely. Yeah, thank you. And Melanie, yeah, it's a very important question. We have to remind our audience here that kinkajou are carnivores, frugivores, I would say, by choice. They arrived in South America several million years ago. They came from the north during the Great Interchange, Inter-America Exchange. I think it's America Interchange. I think it's a good word. And so they moved to south to the tropical rainforest. They were carnivores on the ground, and they have to adapt to new conditions. I mean, there's competition with carnivores, with frugivores on the ground. So they adapted to a new niche, which is the canopy, and night, of course. And so they are consuming what is not consumed by day. And because they are carnivores, they are new in South America, they are quite, I would say, flexible. They are living in very diverse habitats. You can see them close to the road. One of the video I showed was taken like 10 meters from the road. I mean, really, or 20 meters, let's say. It's very close. As far as trees and fruits, they are there. They can adapt. If there's not enough fruit, they will shift to flowers. They will shift to insects. There's some reports in the literature saying that the kinkajou can feed on termites. They have a very diverse diet, so they can adapt. They are carnivores, so they can adapt. We don't know about their real, I mean, if they are eating meat, in fact. This is not so far. So I will say that the role of kinkajou has been underestimated, especially in disturbed forests, as you asked, and in secondary forests, for instance. They might be very abundant as far there are enough resources for them during the lean season, when there's less fruit. If there's not enough fruit, because they are 3 kilos, approximately 3 kilos, they would have a high demand for fruit. So they are important in secondary forest. And I should mention also that even in those secondary forests, they are hunted by people. So they might also be at risk in those forests. So far, people neglect them because it's not easy to hunt them, especially when they are high in a canopy. They don't really call. They don't have much. They are very quiet. In fact, they are very quiet. You just see their eyes if you have a good lamp, and that's it. So most of the people ignore them, and they, I would say, they are quite abundant in the forest. It's funny because once we set the camera in the tree, I mean, really, the first day we arrived, we set the camera. If we go back, and we did that, we go back to several days later, we had a picture of kinkajous the day after, or even during the same night. So really, they are there everywhere in those trees when they are in fruit.
Maybe just a little curiosity to add. Is there any chance of identifying individuals from the pictures? Because, you know, I'm very much interested in individual differences. So if this would be possible, that would open up a whole new area of research.
I think so. I think I will encourage you to view our pictures. There's quite a number of them on Agouti.eu, and I can send you the link, invite you to review them and see whether you can see some difference. For sure, one thing is that kinkajou are territorial. So once you have a kinkajou in a tree, generally it's the same animals coming back every night. Several times during the same night they will come, and then you may have a second individual, another individual, and we do even have a observation of a female, I think it's a female, carrying the small in the mouth and climbing on the branches. So we have a lot of information about the behavior. We have a lot of information about the interaction with the young, with the offspring. And we think that once we set our camera in a tree, we have the information for a family that is from several hundred meters around. And because our trees are really spaced apart, I mean, our protocol at National Road 2 is, I mean, trees are extended along a 30 kilometers road, and trees are apart. Sometimes they are several hundred meters apart. So we may have several families. We may have some interaction between families, but yeah, something to do. Certainly something to do. That's why we have our data fully available for people who are interested in analyzing, yeah, what you mentioned, behavioral ecology, morphology, interaction, anything. It's free.
I think there's many students also here, and yeah, from my fieldwork, I know that you spend most of the time in the field, but it seems like for your work, you spend a hell of time processing the data at home. Maybe Des, you can maybe tell, like, for the new camera trappers, like how does it work to process such an amount of data, and do you use softwares, or do you simply look at hours of video? And yeah, what's the process, the steps?
Yeah, actually, I had to do it twice, because the first time I did it manually just to look at all the pictures as a series, the raw photos. And then I tried to put it in the agouti.eu system just to compare if they would end up being the same. And it's actually easier in the agouti system because they already group them into sequences. So your brain is already conditioned, okay, this is the same sequence of photos, instead of looking at the whole thing as like a big mass of photos. So it's easier in agouti, but it still takes some time. And maybe Pierre-Michel can discuss the new thing that they're working on.
Well, yeah.
Yeah.
Well, thank you. That's well, you could talk about. We are also trying to use artificial intelligence to identify animals that will not be visible to us, especially whether you are a colorblind person, for instance, observing a spider monkey in the green foliage. You may just miss it. The same with birds, for instance. You may also miss them because they are not there. They are hiding in the foliage. They are somewhere far away from your, I mean, you know, it's a flat image. You don't have any depth. So we are going to work with artificial intelligence. we also are testing, and we tested the mega detector. That's something that's been used intensively by people working in North America. Roland Case, especially, I mentioned him. He's a pioneer. He's a pioneer using this system, this software that detect an animal in an image. So we are going to use that too. However, some people recommend not to use mega detector because sometimes, well, mega detector may not see some limbs, for instance, or some ears, some eyes, or some part of animals that are important. And also with our data, you may have some data to be used later that you don't know that are useful. I mean, for instance, movement of branches could be important if you relate that with climate change. So today, okay, there's a lot of branch and leaves and winds and rain. Okay, it's not very interesting whether you are whether you are studying mammals or birds. But for climatologists, it might be interesting. It might be interesting to know whether the trees are moving a lot and to relate that with the wind, the velocity, to relate that with the climate. So we are encouraged to load all images, including those that have apparently no animals. So we probably do that. But then the question is, what are you studying? If you are studying one animal in particular, okay, then you focus on that species. But if you're studying the whole community, as we do, and if you're studying phenology, then it might be interesting to save all those data. For instance, you could start photographing a tree from the flowering period, even during leaf flushing, flowering, and following this throughout the fruit ripening and see every species interacting with flowers and unripe fruits and ripe fruits, etc., etc. And there's probably a lot of information that apparently are not very important for us, but someone else could use it. For instance, we have a lot of picture of animals foraging, grabbing branch. Okay, it's just occurrence of an animal for us. But for someone who is studying the morphology, the handling of fruits, or the handling of branches, whatever, it is worth. So everything is good and everything can be used. And we say in France that everything is good in the cochon, in the pigs. So it's about the same with camera traps. Everything is good with camera traps. I'm sorry for people who don't eat pigs. Anyway.
It's a very rich data. Do we have more questions? Please raise your hand or write in the chat.
There's a question in the chat to Desamarie, and the question is, at how many night traps did you inventory all the carnivore species by Niana?
The number of trap nights is 1,812 for this season. So a little bit more for the next season because I'm adding a few more camera traps.
And to complement, we have studied trees for 2 months in average, more or less, during the fruiting season, early season, and we've done it November and December. But now we're going to start a new project where we're going to leave the cameras for 1 year, entire year, and for several years. So that means a lot of information to save and then to analyze. And that means a lot of people to analyze them, to work with them. And a lot of camera, a lot of travel, etc., etc.
Thank you. Any more questions? We're coming also to the close of to the end of our talk series. So your last chance, or not your last chance. I'm sure Pierre-Michel and Des are also okay with you writing them in person.
Well, thank you everyone for listening. Thank you for Des. Thank you to Des for contributing. And unfortunately, I was not able to ask a third person to present her work, and I thought to Jessica Isom, who did a really wonderful work in Sabah. And I hope that we can have her sometime and some other like Tremi or Jennifer Moore to speak more about camera trapping, because really, I would say these women are really strong.
Maybe you two can give like an end remark. How do you envision the future of camera trapping? Because I mean, the technology is developing so quickly. Do you think that in the future it may not be used anymore, and we focus more on DNA barcoding, or do you think it's really like developing and will take over a lot of other methods used in tropical ecology, or what do you think?
Well, no, I don't think. I mean, all methods are complementary. DNA is used to complement what we observe. It's always the same. The same question is to observe what we cannot observe. I mean, to measure what we cannot see and measure directly. So the camera traps is exactly for that. I mean, it's not because you set camera traps in a tree that you have all the community from that is foraging into a canopy. No, we are missing a lot. We are missing some small animals that are using the branches, the edge of the trees, because we cannot, even if it's fancy to do it, but we cannot really install camera traps at the tip of the branches. And you have a lot of birds and small animals that are foraging there. So camera traps are just a complement to other methods. That means you still need to look to observe them with binocular, but for sure at night in a canopy, it's really complementing the work on the ground. And I would say final word is really train, train, and train. Train to climb, train to set the camera, use camera, and then train to analyze the data, because there's a lot to take from those data, a lot.
Yeah, I think I agree that camera traps are a little bit irreplaceable, but maybe in the future, camera trapping will change from the way we know it, because right now the most dependable camera traps are made by corporations or like big companies that and they're expensive. So maybe in the future, when we have this whole generation of techie kids that can make it cheaper and with 3D printing and everyone knowing how to code, maybe we can make it more cheaper and more widespread in use in the future.
Great. Yeah, and you will be part of developing this field further. It was a really nice talk today, and we thank everyone who participated and asked questions and listened. So just a few remarks about when the recordings will be available. So they will be published on our YouTube channel, and you will also get information on our website under conferences. So here you can also see all the upcoming events listed. And for our next talk, we will have in November on the second Monday. The topic is thermal tolerance, and after that, paleoecology. So you're again invited to join. And also, if you want to present or propose someone to present, you can write us. You see the email in the lower right corner. And if you want to be part of the email list to get like the links of every month's Theo Talk series, you can also write us to theo@soctopecol, and we will reply you and put you into our email list, so you're always up to date what's going on with the Tropical Society.
And
right.
Join the society.
Yes, join the society. It's easy. All right. Thank you, everyone.
Thank you, everyone. See you in the
Have a
next
good
meeting
week.
and next